welcome to our fifth vodcast in the information domain. In this one, we're going to be exploring cell cycle and mitosis, and we're going to start to look at how cells make new cells. So the big questions: why do cells need to divide, and how does the division provide for continuity in terms of processes in individuals versus the propagation of a species? So without further ado, so why do cells divide? Continuity, right? We know that from cell theory that cells come from pre-existing cells. So in order to get more cells, we need to go through division. So the individual, as an individual, we have to replace cells that are worn out, damaged, in terms of growth, general maintenance. These are all reasons why we need to promote and have new cells. So when you're growing, like when you're an itty bitty baby to when you are now, your ratio of new cells to cells dying needs to be way greater. So you need to have more cells being made than die. Okay, and that's how that works. So, however, in order for the species to survive, species have to replace individuals. So cell division also helps to accomplish this. And when we get into talking about particularly in a single-celled species, yes, mitosis is going to be one of the main ways, or binary fission, whatever you want to call it. And then for sexual reproducing animals or organisms or whatever it is that uses that, because it's not just animals, cell division is also an important process to help achieve that goal as well. So here's the cell cycle in all its glory. The phases of the cell cycle are very important, and you're going to hopefully have some, your brain's going to be like going back saying, wow, yeah, I definitely remember this. So take a second to peek at it again. Major parts to the cell cycle are interphase, which is broken up into three parts: G1, GS, and G2. G1, general growth, general cell doing its thing. You're going to have the G0, which would be a non-dividing state, so the cell basically maintains a constant G1 phase and won't ever redivide again. This is a lot of cells in you will do this. They'll never actually divide once they hit an adult phase. The S phase, you're going to get your DNA replication, G2, prepare for division, and then you have mitosis, which is the actual division of the nuclear material. And this is really, really controlled. So let's, you want to be thinking of some reasons why it's going to be so controlled. Well, chromosomes, okay? And we know that chromosomes are a big piece in a cell dividing, is that we have to make sure that the new cells, our daughter cells, contain the correct chromosome count, depending on what it is. So chromosomes are extraordinarily coiled pieces of DNA condensing prior to division. So you got your long piece of DNA wrapped around histones. They're going to continually wrap, and in the S phase, they are going to replicate and then connect at that centromere. So for most critters, like us and a lot of eukaryotic cells, there's two copies of every chromosome because we got two parents, right? So, however, remembering the fact that prokaryotes are kind of over here in this discussion because they only have a singular circular chromosome, okay? We have a lot of linear chromosomes. So for prokaryotes, their stuff's a little different. They're going to replicate that circular one. They're going to go through cytokinesis divide. That's binary fission. It's a little different than the processes of mitosis. So just to kind of keep that in mind. However, there's some terminology we need to make sure you get squared away, okay? So you got to understand the difference, because as we're talking about them, really what we're talking about are different moments in time, okay? It's really the same thing. It's just semantics, right? But we got to understand what's going on at the time we discuss it. What a chromosome is, okay? Piece of DNA, particular gene placement, length, all that jazz, right? You're going to have the centromere, which is a unique location on every chromosome, and then you have your P arm and your Q arm. Just so you know, the P is usually the shorter part, so whatever part's shorter on the chromosome, your Q is going to be your longer. Then you have chromatid, and what a chromatid is. The only time you refer to a chromosome as a chromatid is when it's in its replicated state after the S phase. So you have a single chromosome that replicates, and when it's attached at its centromere, okay, you've got a chromatid, and then you have its sister, okay? So that would be chromatids. And then in mitosis, when those chromatids split and divide, they go back to being two chromosomes again. It's the same thing. It's just what moment in time are you describing it. So keep that part going. More terminology as we get more into our discussion. Again, this should be dinging off bells in your head because you have heard these before. Haploid versus diploid. A haploid count, any cell that is haploid only has one of every chromosome type. In essence, half. If you're a critter that has two of everything, it would be half your chromosome count, one of every type. So that we give it the variable designation of N. Diploid, you got two of every type. Most of the cells in our body, with the exception of one cell, are diploid, okay, which means we have two of every type. Think of it like types of socks, right? Different pairs. If I've got footy socks, I got tube socks, I got ankle socks, I got toe socks, they all come in pairs, right? So there you go. That would be my 2N. The N would be the type of sock. I have one footy, one tube sock, one toe sock, one ankle sock, one sock with the little fuzzy ball on the back side. I think you get what I'm saying, right? We as humans, we've got 23 pairs of chromosomes. Okay, again, pairs. Why? Because we got a mom, we got a dad. Okay, and that's how it works. So how many chromatids then, chromatids, are present during G1 and G2? So you kind of have to ask yourself that as we're going through some, kind of jot that down as you're going so that you can understand what that's all about. So here's the big thing. We're going to focus on mitosis here, non-reductive eukaryotic cell division. What that means is I'm not going to reduce the chromosome count. I'm going to start with a 2N number. I'm going to end with a 2N number, whether I'm talking about us, whether I'm talking about I don't even know, slime mold, blue whale, whatever you want to think of. Anything that's going to need to create more cells is going to go through this process. So we begin with this non-reductive. We're going to keep and maintain chromosome count. Interphase, that's what I mentioned before, your G1, your S, your G2. Growth, replication of DNA, prepare for division. You're never going to notice a cell in interphase in terms of which one it's in. You can't tell yet, okay? So here are just a couple different examples. You got newt, whitefish, and onion cell. There's interphase. You can see the nucleus. The nuclear membrane is still present, okay? And the internal, you might see the nucleolus like you can in that onion cell, and various other pieces, okay? So there's that. Once it's signaled to begin the mitosis phase, now we move into prophase. Here is where your chromosomes are going to begin to condense. You're going to be able to see them like you can here with that real kind of, you can see them all kind of wound in there. You can see them all over here. You can see them all here, but you get that condensing piece. The other piece to being able to identify is that the nuclear envelope breaks down. You're not going to see that fine line that designates the nucleus anymore. The spindle begins to form, okay, which is in animal cells going to come off of centrioles. In plant cells they won't, but the spindle is the series of microtubules that these chromosomes are going to attach to at their centromere in order to, it's like a track. They're going to put them on there and they're going to move from that. So that begins to happen in prophase. You can designate based on how much the nucleus is gone, how condensed they are in terms of, you can have sort of an early, mid, late prophase within there. So here's prometaphase. This would be like that midpoint between prophase and metaphase. This is where things are really starting to happen. Chromosomes start to migrate towards the center. You get two very complete spindles now at poles. Okay, so the spindle fibers are going to move so that they're at opposite ends from one another, creating those strands. Okay, that's prometaphase. Once you're in metaphase, the chromosomes are literally down the center of the cell, right down that equator where they're going to then divide. Okay, so you've got that. They attach right at the kinetochore of the chromosomes at that centromere, right onto the spindle, lining right up down. Because the key is, if I have two chromatids attached, right, I'm going to pull them apart. Again, just difference in terms of terminology. Both together, that is one chromosome. Two, like one, I would say, replicated chromosome with one chromatid and its sister. Those sister chromatids are going to be yanked apart from one another. So there you can see kind of what's going on there with the kinetochore at the centromere. Okay, microtubules coming off of it, and what's going on with the actual chromosomes sitting there. And all the rest of them, their arms are just kind of dangling out there. Then anaphase. Anaphase is where the actual chromatids split apart and you get migration towards the poles. Okay, so you can see in various stage, basically different options. Again, that's what these are. Same critters throughout, the newt, the whitefish, the onion. And you can actually showcase and see the chromosomes being pulled apart at the centromere, which is pretty cool. And the way it works is that they don't like travel on the track. It's actually the pulling of the microtubules actually yanks them, okay, towards one end, which is pretty cool. And then in telophase, this is going to be sort of telophase and cytokinesis are kind of happening all at the same time. Chromosomes are on either side now. They've been pulled apart and separated. They're going to begin to decondense now and kind of, re-unwind. You're going to get the formation of the new nuclear envelope around them, and cytokinesis is going to occur where you're going to get the splitting of the cell. In an animal cell, streaming is going to happen. If streaming happens this way, animal cells going to begin to do this, and you're going to get formation of a cleavage furrow as that pinches in until, voila, you get two cells. In a plant cell, that can't happen because of the cell wall. You get literally a formation of a cell wall right down the middle, and we call that the cell plate. And eventually that forms so fully across the cell and is so up and down and across that you get two very distinctive cells as a result. So that's cytokinesis. So here again is that difference between the splitting of the cell in an animal and the splitting in a plant. That cleavage furrow, okay, as it pulls in, it's a contraction ring of microfilaments actually will pull and yank, okay, as cytoplasm streams outward, pull that cell membrane down, and you get your two daughter cells, okay? However, in a plant, like we said, can't do that. We form the cell plate, and then how the organelles separate, that's kind of random, to be honest. Okay, so there's your animal, and you got an SEM cleavage furrow showcasing how that works. Plants, that's how it works too, building up that cell wall. Cool. So here's kind of mitosis. Mitosis at a glance. Here it all is in one sort of event. Okay, so there's that. Now, spot the phases. Okay, designate each row. So we've got row 1, 2, 3, 4, 5, 6. Okay, and then you've got 6 rows, and you can going from left to right which cell. So in row, okay, I want you to find and write down in your notes, or actually we'll do this, we'll put it in the whisk, okay? So in your whisk answer, okay, you're gonna tell me how many cells on here, how many cells and at what location, okay, are in anaphase. And then you're gonna tell me the same thing for how many cells are in prophase. So again, prophase, how many cells in what location, and anaphase, how many cells and in what location. So that's your practice right there. So you'll answer those in your whisk question. So going, okay, so again, from here, jump into the original, just the link to this to actually watch these, and you can actually see video of cells actually dividing, which is pretty dang cool if you ask me. So you'll be able to see them. Now, mitosis has gone through a series of sort of evolutionary events, just like kind of all life has. So more similarities between eukaryotic and prokaryotic division than you might really get kind of at first glance, because they're both binary fission, the idea that you're creating two identical cells. Okay, so the proteins that are involved have a very large degree, the meaning like the microtubules and things like that, have a large degree of homology. They're very, very similar in their construction, in their nature. And so by having those, that's sort of like a thread that kind of holds the idea of how life has moved forward. Some unicellular eukaryotes are going to demonstrate sort of these intermediate modes of division, similar to something like an amoeba or a paramecium. So just kind of take a look at that variation. So with that being said, don't forget to make sure you can do these things and that you know the phases and you know what happens during each phase. And be thinking about those as we work our way through. All right, so with that, have a great night, and we'll catch you guys in class. Take it easy. Bye.